had offered 1.5 million for this one.
No, but he is pretty good at his thing. So we've been working hard on trying to make it.
Morgan has been a fixture in the MVZ and has been a major contributor to the Grinnell Resurvey Project. But to give him due introduction, Morgan comes from, let's say, relatively impressive background. Got his undergraduate degree at Harvard, one of those small schools. A master's at Oxford before joining Steve's lab here at Berkeley. And as many of you know, he has really been the Avian component in a lot of ways of the Grinnell Resurvey Project. That he's written, as part of that, a paper that's emerging as a very seminal, important paper. It was published in the Proceedings of the National Academy of Sciences on avian changes in the Sierra over the last 100 years. I suspect we're going to hear a little bit about that today. I didn't ask Morgan if this is almost your finishing seminar or not. Should I ask that question? Or is that
I asked Steve about it, and he says you have to be finished to give it. So I still have one semester left. But it's probably as close to an actual defense as I'll come.
Okay, so you guys are here for that moment. But anyway, I'll let Morgan take it away. Today he'll be speaking on 100 years of avifaunal change in the Sierra. Thanks, Morgan.
Thanks. So, I've been a grad student for 6 years, and I'm always surprised by sort of the lessons you learn every time you give a presentation. No matter how many presentations you give, there's always something that comes up. And it's kind of like sitting down to sort of have a good photo taken of you, and suddenly having a old man ground squirrel pop up in the middle of the frame and try and ruin your photo. And I found that sort of happened this week because I was, a week ago I was ready for my presentation, and then my laptop crashed a week ago. And I took it into Apple, and they said 3 days, and I didn't get it back till yesterday at 4 p.m. And so I was really convinced, actually, that I was going to be giving the first modern MVZ chalk talk. But lucky for you, that is not the case. My research, as many of you know, is basically all founded on Joseph Grinnell, who really should be a co-author on all of my papers. No MVZ Grinnell talk would be appropriate without basically a good photo of Grinnell, and one of his prescient quotes, talking about how we should really be taking the best notes that we can and collecting as much data as we can, because we don't know what use that data will come to. And we're sitting in the room that's sort of in honor of that. And so I've had really the luck to sort of go back and sort of dig around in Grinnell's journals and sort of really walk where he walked and redo what he did. As a side note, as part of that, I've discovered this is actually not the first photograph that they took in this series. This is the second photograph. The first one was actually ruined. So just a general outline of my talk today. I'm going to be giving some really basic background to the Grinnell Resurvey Project, because I'm sure everyone here has actually probably heard at least something about it. Then talk about sort of what we found in terms of elevational movements in Sierra Nevada birds, and then finally looking at sort of trying to understand those movements from a more climate perspective. So when we think about Grinnell's legacy, often in the museum here, we look straight at all those cases and look at the specimens that he's collected. And certainly Grinnell and his colleagues in the first 40 years of the 20th century sampled a whole lot of California and collected a whole lot of bird specimens. But the focus of my dissertation is actually over here on this side, and just what's come out of the field notes. And Grinnell really was ahead of his time in taking pretty systematic bird surveys. So he created this census sheet and sort of decided that he was going to start marking sort of hour-long periods of time. And in this case, he was climbing Lassen Peak. And so the elevational bands he'd be in during those hours, and keeping a full tally of the numbers of individuals and species he found. And so he did this, and this is another example from Tracy Storer in Yosemite. Sometimes they didn't have the field pages at hand, so they just kept a list of the species and did tallies. And sometimes they did the tallies sort of on another piece of paper and then transcribed it into the field notes. But I've been able to go back into the field notes and sort of extract out all these surveys and be able to use them as a historical baseline that I can go back to and resurvey and resample to look at how the bird species and bird communities have changed over the last 100 years. This project started before I came, and it really started in 2003 with the first Yosemite project, going into Yosemite and doing samples of mammals, but then birds. And Andrew Rush, who's here, right? saw him come in the back. Andrew pioneered this in Yosemite in 2003 to 2004 when he was an undergrad, before he left us and then came back. I came on in 2005 and started my work up in Lassen. In 2006 and 2007, we expanded down into the southern Sierras and what we call the Whitney Transect in 2008, 2009. We also did a small bird survey, resurvey project along the I-80 corridor in 2008. Peter Panchin, who's a postdoc, is now doing bird surveys on coastal California, and I think that's probably continuing this summer, Pete?
Yeah, all the data's collected.
Oh, it's all done now, 2009, 2010. And then also a former field assistant of mine and someone we all know well, Alison Schultz, who's an undergrad here, did a resurvey of campus in 2007 to 2008. A side note that I'm not going to go into a whole lot, but it's really important to how I actually analyze my data, is how we compare our two data sets. Because we have our sort of modern bird surveys taken sort of a modern methodology, and we're trying to compare it to Grinnell and what he did in his time. Because the methods are slightly different, and any time you're comparing basically observation data, you have this issue of false absences. Where you have species, or you have basically a species that you did not find at a site. And how do you know that actually the species was not there, a true absence versus a false absence? The species was there and not detected. And so there's a method that's been developed over the last 5 years or so known as occupancy modeling, where you take repeat surveys at a site, and you use this to calculate for a species in a site a probability of detection. So you can model this probability of detection, and you can use that probability of detection to conditionally model the actual true probability of occupancy of our species at a site. And this modeling framework has a lot of flexibilities, and one of the powerful things about it is to be able to relate different covariates to both the probability of detection and the probability of occupancy. And so, for instance, we can test for whether or not there's a difference between time periods in the probability of detection of a species, and also the difference between time periods and the probability of occupancy. We can look at how the probability of detection changes for a species over the course of a breeding season, as singing frequency and activity changes over the course of a breeding season. And then we can also relate the probability of occupancy to a lot of other factors related to questions we want to answer, such as how occupancy changes over an elevational range, how it changed over time, and how it can change across our 3 survey regions between Yosemite, Lassen, and the southern Sierras. And so in the sort of analyzing of my data, I've taken a bunch of different detection models, and I've taken different parameterizations of occupancy, and I've modeled them all on a whole bunch of species, and you combine this into a single sort of averaged model, which will relate in some ways as this probability of occupancy along an elevational range. And so we can model this sort of the true occupied elevational range of species and how that varies over region and over time. So I've done this for a total of 114 sort of pretty abundant species found in the Sierra Nevadas. And to summarize what we've shown. Before I go into my results, I want to talk a little about sort of what we actually know about birds and climate change. And I'm going to keep this pretty brief. But a lot of the studies that have been out there and that have published looking at avian response to climate change is phenological. We have evidence of species migrating earlier, of species nesting earlier, of species fledging earlier, and that's where a lot of the research has been focused. The other flip side of that is the expectation that as temperature is changing, species are going to be changing their ranges. There's been some good evidence in Europe and in Europe and Britain of bird species shifting northward and latitudinally with warmer temperatures. And there's been at least 2 studies across North America of species shifting northward, but that's also only for the winter range. While we expect species to be shifting north, we also expect them to be shifting up in elevation. We don't have great evidence in this case. The best examples that we have, particularly for birds, are from tropical locations. Because of these difficulties in actually having long-term research studies, very few of our time series are longer than 30 years, even though we get decent signals in certain cases for 30-year time periods. And across all studies, there's a problem, particularly in elevational areas, of getting replication over any sort of spatial scale to know that what you're seeing locally is not just a local effect, but is actually a regional or continental effect. So to sort of to get at this, the data set that Grinnell has provided us sort of actually allows us to compare across 100 years and across 3 different sort of elevational cross sections of the Sierra Nevadas. So I've already talked about these 3 regions, but this is sort of the foundation of my research, looking for species shifting upward in elevation. And sort of, you can look at sort of one case and one example and see just kind of what that data looks like. And so here we have these dot plots of both the historic time period and the modern time period for our 3 regions. And with this example of the mourning dove, you can see that its upper elevational range has shifted up. In Lassen, it has shifted up in Yosemite, and then shifted up in Whitney in varying amounts. And this is what you get out of occupancy modeling. This is the historic curve, and in the brighter, lighter color, the modern curve, showing that in each of these cases that modeled true occupancy has shifted up. And we can look at one species, but looking at one species doesn't tell us all that much, because we can't actually prescribe any causality to it. We can't say that these didn't shift up by any other response. It might be temperature, or it might be something else. So for that, we really want to look at sort of aggregates of species and look at species groupings as a whole, and see what all species are doing to see if there's any sort of pattern that is consistent across all species with climate change. And so when we're looking at range changes, we can have sort of a hypothetical mountainside here with a hypothetical species that's distributed across. And as we model that, you might get this expected curve where the probability of occupancy relates with elevation, and this is the area where the species occurs the most. And then we can look at 3 different parts of how the range might be changing. We can look down here at the lower elevational limit and see how this changes over time. We can look at the upper elevational limit, and then we can look at the middle. In this case, I've analyzed it by using the median of the cumulative occupancy curve to basically try and get an idea of how the distribution as a whole, and perhaps the distribution where the most individuals are, has changed. So I'm going to present results for these 3 different areas separately. Looking first at sort of this median occupied elevation, or what's happening in the middle of the range. As you look across these species for all 3 regions, and these graphs are the fraction of species in each region that have shifted up, shifted down, or not moved at all. And when you're looking at this range as a whole on aggregate, you can see that we have this really strong signal of species shifting up more than they are shifting down or not moving. And this is what we'd expect, and this is consistent with what we find in other parts of the world and other studies. You can see that there is some variation. Whitney certainly doesn't have as strong a signal as Lassen and Yosemite, and that'll become clearer as we look at the other range limits too. But when you take all these species in the 3 different regions and you put them into a mixed model with species as a random effect, you do get this coming out that across all 3 regions you have this significantly more movement sort of up in elevational range than downward or no change. If we look at the lower limit, so the lower range limit can either stay the same over time, it can shift down, which is mean range expansion, or it can shift up, which would be a range contraction. I would split these up in terms of the species that are found, that are basically low elevation species and high elevation species. Because we found that species, these bars actually shift very differently depending on which of these 2 groups you're looking at. So if you're looking at the low elevation species first, these light green bars, pretty much low elevation species are not having the lower elevational limits change at all. 1 here, 0 here, 2 here, 7 there. There's not very much happening for the lower elevational limit of low species. High species have a strong pattern, particularly in Lassen, of shifting up. When we put this into a mixed model, we don't have any significant effect of region, but we do have this elevational zone significant effect, showing that there's something different happening between low elevation species and high elevation species. When you look at upper range limits, again, your upper range limit can expand, it can move up, it can contract by moving down, or it can stay the same. And you look at across the 2 elevational groupings of species, we find that that low elevation species are actually moving in this case. So low elevation species are expanding up and they're shifting their upper elevational range, while high elevation species aren't doing that at all. And this pattern is very strong in Lassen, in Yosemite, and it's slightly there in Whitney. And when we put this into a mixed model, we get the significant effect of both elevational zone and region, particularly differentiating Lassen and Yosemite from Whitney. And so we look then at sort of median range, upper limit shifts, and lower limit shifts. We find that on the whole, species are moving their entire range up in elevation, although there's a fair amount of variation. Low elevation species are expanding upwards, and high elevation species are contracting up. And that's the general pattern we get, but there's a whole lot of variation. The next thing we wanted to look at was, we have all these species that are acting different ways. Is there any sort of predicting of, of a life history correlate with whether or not a species has shifted its shifted its elevational range up? And so we collected data on, I collected data on body mass, home range size, clutch size, the territoriality of species, and then two types of migratory variables. One was whether or not the species was a year-round permanent resident in an area, and the other was whether or not it was a broad neotropical migrant. And when we put this into our models and tested it with the median elevational range, or sort of the range-wide, whether or not the whole range had shifted, the only predictor that actually improved the model at all, and only marginally so, was this neotropical migrant status. In the fact that whether or not, when you're looking at whether or not it's the likelihood that a species would actually move upward in its elevational range, migrant species, neotropical migrants, which have much lower annual survival, much more turnover, and are certainly far more badgeal in many cases than resident species, have a higher probability of shifting their range up. And that might be what we'd expect a priori. At least that's the case I would make. That's the situation that we'd expect. So this is a encouraging result in terms of life history correlates for this type of behavior. When we looked at upper limits and lower limits, looking first at lower limits, we didn't find any predictor that actually improved our model. So we didn't find any life history correlates that were a significant predictor of whether or not the lower limit would shift up. When we looked at upper limits, we only had one, and it was a very strong predictor, which was clutch size. And you might expect, we expected, that higher clutch sizes would be more likely to shift up in elevation because they're producing more young, and you have more species expanding instead of moving off territory. And that's actually not the effect we have. And so this is the results for both high elevation species and low elevation species in our three regions. So if you look at these two top lines, which are low elevation species in Alaska and Yosemite, you can see they demonstrate this pattern where low clutch sizes actually had the highest probability of expanding their upper limit or shifting up in elevational range, whereas high clutch sizes had lower probability. And I don't have a good explanation for this, but it's what we found. So that's a description of what I found in terms of how species have moved through elevational ranges. But that's sort of on aggregate looking at all species. And what I found sort of when you start looking at individual species is you start seeing some interesting or sort of odd behavior that comes from the different regions. So if you take an example with the white-headed woodpecker, in Lassen the species is very, very consistent with our climate change hypotheses. Its upper limit has shifted up and its lower limit has shifted up. In Yosemite, its upper limit has shifted down and its lower limit has shifted down. And in Whitney transect, its lower limit has not changed and its upper limit has not changed either once you account for the probability of false absence. So we have one species that is doing three different things in our three regions. And how do we make sense out of this? And a lot of what I've been struggling with in the last few years in my dissertation is trying to make sense out of this when we expect all species to be responding the same. And I think a lot of it probably has to do with climatic variability and how climate has changed differentially, even within one region like the Sierra Nevadas over time. And so this is a plot in climate space with temperature on the x-axis and precipitation on the y-axis. And each of these arrows is one of our sites where we surveyed in our three regions, and I've added in I-80 for this portion as well. And so the arrows take you from where the site was climatically, historically, in breeding season, temperature and precipitation, and where it is now. And you can see that there's big differences both across regions and within regions. So Lassen, for instance, these green arrows has seen huge increases in precipitation relative to pretty small changes in temperature, whereas Yosemite has seen really great increases in temperature, particularly at the highest elevations. And the southern Sierras has really had no change in precipitation because there's already a dry zone to begin with, and has had, in some cases, some pretty extreme warming. So if you take all of this in, you might have different predictions or different expectations about how species are going to be responding, particularly on a site-by-site level. And so in thinking about this, I got taken back to Grinnell. And a lot of this work was spawned because of the Sackler Symposium that happened almost 2 years ago now. It's some work that I started with Craig and Steve and Bill Monahan, who was still around then. And so we got thinking about how Grinnell defined species ranges and how species, and really correlating, relatedly, how species ranges might change. And so his seminal paper, where he defines the ecological niche, which then became the Grinnellian niche, was talking about the California thrasher and the factors that relate and describe the California thrasher's range. And this got us thinking about how species, these ranges, changes that we see, and changes in the occurrence of species at sites over time in these areas might be related to the ecological niche of species. So we could develop this framework where we can think of this big circle as the environmental niche for a hypothetical species. Within this circle, the species does well, thrives, has a positive growth rate, and outside of it, it does not do well. And then we can have a geographic space that we actually have that exists, and a certain portion of the geographic space intersects with the ecological niche for a species. And if we then sample it within that geographic space, where that geographic space intersects with the niche, we expect to find the species, and outside of the climatic niche, you do not expect to find the species. And what happens with climate change, just like those arrows that I showed earlier, is that each of these sites is moving through climatic space and time. And the question then is, what is happening to the fate of that species at those sites? If the climatic niche is not actually impacting where the species moves, then you expect the occurrence to actually stay the same, for these two sites to stay occupied even though this site is no longer, or expected to be no longer climatically favorable, and this site to remain unoccupied even though it is now climatically favorable. If the climatic niche is actually important in driving these occurrence changes, then you'd expect one site to go extinct and another site to become colonized. In this latter case, we describe as is defining niche tracking, where species are following these limiting environmental boundaries through geographical space in order to remain in favorable climatic space. We wanted to try and test this with our species and with the patterns of colonization and extinction that we found, but in order to do this, we actually had to come up with some sort of measure of what the climatic niche for a species is, which is not a trivial thing to sort of try and think about. The way we did it was we took historical specimen records across the entire ranges of Western distributed North American species. So we went onto ORNIS and we went into the MBZ, and we took out all the historical specimen records prior to 1940 for 53 species. Of course, we took only the ones that were georeferenced and in the breeding season, and we plotted them, like here, for instance, for the Lasley bunting. Then you can take these and you can plot them in climatic space, and then use this to really describe, in some ways, is what your climatic niche might be. And we decided, since we don't really actually know that much about the boundaries or what a boundary means in this case, the best description of the climatic niche that we could come at would be the centroid. So we can take that centroid and we can plot it in relation to where the species was found historically, in these blue dots, in the Grinnell Resurvey Project, and then where we found it in the modern resurvey work. And then those can also be plotted by centroids. And then you can really then just focus on the centroids and look at the relation between them over time. In particular, if this is where the species was in the Sierra Nevadas historically, and this is where it is now, what's the relationship between these two, and how is this moved relative to this niche centroid? And what we decided was that it wasn't maybe necessarily how close it is now or how far away it was then, but really what direction it was moving. Is it moving in the same, are these vectors, the vector for how it actually moved versus how far the vector from the distance between the historic centroid and the niche centroid, are these signs the same, and are they moving in the same direction? So in this case, with the Lasley bunting, we found the species both sort of historically existed in an area that was warmer and drier than it was than the niche centroid. And then over time, it moved in the same direction. So in this case, there would be evidence of tracking, for tracking both temperature and precipitation. And now we used this framework to measure 53 different species, and found, as this table sort of demonstrates, that sort of pretty much all but 5 of the 53 species were tracking at least one environmental gradient, either precipitation or temperature. And only 5 showed no evidence for tracking of either. And what we looked, one of the things that struck us about this, and an unexpected result, was you look at these 5 species, all 5 of these species are species that are urban adaptable, and sort of can be classified a priori as urban adaptable. And as if you wanted to go on sort of a thought experiment and say, well, what species might not be having their geographic range impacted heavily over the last 100 years by climate, it might be species that can escape climatic variation and escape climatic pressure by taking advantage of human-dominated landscapes. Another sort of thing we found that we did not expect was looking at whether or not species were tracking temperature or precipitation. So if you look at this, these species are actually ordered in terms of their average elevational range. So low elevation species found that their range was pretty much entirely dominated by tracking precipitation, whereas high elevation species were tracking temperature, and a lot of species in the middle were tracking a bit of both. This is obviously a significant pattern, and it's a question of, it's a pattern we don't really know how to explain. We know in some cases that if you look at what limits net primary productivity as you move up in elevation, in the lower elevations, NPP is limited a lot by precipitation. In the high elevations, it's limited by temperature. So this might be a signal that the species are actually tracking something entirely different, and that they're not responding directly to temperature and precipitation. They might be responding to something like NPP. But it's certainly an intriguing pattern that we found. The last step with this that we wanted to look at was we wanted to say, okay, we have this evidence of species tracking temperature or precipitation, but how does this actually relate to how we, in some ways, how we predict how species respond to climate change in the future? So to test that, we went back to these historical full breeding range distributions, and we modeled them using Maxent. And we took out of Maxent, we modeled them in Maxent using only temperature and precipitation of the breeding season. And we took out of that basically whether or not the climate envelope was best contributed was most strongly defined by temperature or precipitation, to come up with a priori estimates of whether or not the full range of a species was best described by temperature or precipitation. And we compared that to whether or not the species empirically had tracked temperature or precipitation. And we found, again, really striking, very close relationships. The species whose full breeding range was best described by temperature on a large continental scale were also the species who were tracking temperature over time. And the same is true for precipitation, which gives kind of an encouraging note to people who are actually modeling future ranges of species and trying to see what the future impacts of climate change are, because it means that we might possibly be on the right track, even though we can't validate our models of how species are going to react in the future. This is giving some indication that at least when we do niche modeling and we use Maxent, we're actually pulling out environmental factors that may actually be meaningful, which is kind of encouraging. The last step now, and we've been using centroids and movements of centroids, but I mean, really these movements of centroids are just the manifestation of what's happening at the actual individual sites in changes in occurrence over time. And so the last step that I wanted to do was really to sort of go back to these actual occurrence patterns and see if we can sort of detect a signal of the niche and the way that niche is driving driving colonization extinction in actual sites. So to do this, I had to build a separate modeling framework. And in this case, we have our niche centroid here, and then we imagine our groups of sites that we've sampled around it. And so theoretically, we expect these, the closer you get to the niche centroid, the more likely, then that's where the species is going to be, that's where the species occurrences are going to be lumped. In a null model, we might expect that over time, if you look at colonizations in green or in extinctions in red, a null model would expect that those patterns of colonizations and extinctions would be random, at least in relation to where the location of that niche centroid. In a separate model, though, what we call our static model, how far you are from the niche centroid determines whether or not you go extinct or become colonized. In this case, the closer you are to the centroid, the more likely you are to become colonized. And the farther out in the periphery you are, the more likely you are to go extinct. Our third model was a model that incorporates the idea of climate change, and that the fact that these sites are moving over time. In this case, if you started out really far from the niche centroid and have now moved closer, you are more likely to become colonized. And if you started out, however far away from the niche centroid, but you've now moved very far from the niche centroid, you are more likely to go extinct. So we put these three conceptual models to the test, going back to an occupancy modeling framework and using a two-season model, where we model, in this case, the initial historic probability of occupancy, and then we model the probabilities of extinction, the model probabilities of colonization, and we can look at what factors best describe the extinction and colonization probabilities. Our null model just has an intercept which says that that's colonization and extinction is random. Our static model uses just how far you are away from the site is from the climatic niche historically. And then our dynamic model, which we did in two parameterizations, uses both how far away you were from the climatic niche historically and how far you are away from it now, in the relations between those two. So we took these three different parameterizations and we tested them for our 53 different species. And then for each species, you average across all models and calculate the sort of AIC, cumulative AIC weight for each of the models. And then we've averaged across each of all 53 species to get this table. And so these are sort of weights and evidence in support of these species, averaged across 53 species. And what you see is that sort of overall we have very, very, very little support for this null model. And then the support was really split between the static model, which is just saying that the niche is important for driving colonization and extinction, that how far you are away from the niche, and the dynamic model, which says that the niche centroid is really important, but it's also important how much you've moved since then. So this is the only one that actually incorporates the idea of climate change. But altogether, this is saying that this location of the climatic niche is the best description we have at actually explaining the individual site dynamics and colonization and extinction at these sites. So, some final thoughts to sort of wrap up all that I've presented. I mean, certainly we found that birds' ranges have changed a lot. If you look back at our tables, at those graphs of upper limits and lower limits, you have this signal that species are actually shifting up in elevational range. But there's a ton of variability. You have a lot of species that have shifted down in different portions of their range and their entire range. And you have species that have not shifted at all. So, in some ways, when we're looking for climate change impacts, we can see this signal, what Terry Root and others have called sort of the fingerprint of climate change. You can see it in our data, and certainly I think it's one of the few sort of large regional elevational breeding season studies to do that. But there's a whole lot of other variability there that really shouldn't be ignored. And I think the second part of my work has really sort of highlighted the fact that when we're thinking about climate change, we need to be thinking about more than just temperature, particularly with birds, but I think with all animals. There's this idea that maybe temperature is the only thing that's driving climate change. And I think in California, where we have such extreme precipitation gradients and high variability in terms of how that may be changing over time, we should be focusing on that a lot, more so than we are. And in some cases, when you look at a lot of these studies, when you look at individual species, you look across and you say, oh, species are behaving individualistically. There's no framework for how species are responding. And I don't have an answer to that, but I think what I take out of my work with the climatic niche studies is that species all have their unique individual characteristics, and they're each, as Grinnell described, they're each psychological and physiological things that are describing where they are, and that might be really coming into play how species are shifting their ranges over time. And then the trick then, as we're looking forward, and trying to understand how things are responding to features is something that I think Grinnell would approve of, which is the challenge is in actually trying to figure out what that niche is for each species and how we might incorporate that into understanding the future. And this is not a trivial thing at all. I mean, we simplified the situation a lot by calculating these niche centroids, and it worked out, but certainly that's not going to help us that much in the future. And so the real challenge lies, and we're making a lot of progress in it, but the real challenge is best describing that niche and using that to really understand current distributions, past distributions, and future distributions. Since this is close, I'm going to get to a dissertation defense. I'm going to excuse me for having like an Academy Award moment. A lot of people to thank. Foremost, Steve Beisinger, my main advisor, and the rest of my dissertation committee, Craig Moritz, who's been the big leader all these years in the Grinnell Resurvey Project, and then Rory and Justin Verschares. I've had a lot of really great field assistants, although I really call them field leaders. Andrew Rush, who was not even an assistant of mine, but led this project from the start in Yosemite and allowed me to use 2 years of his data for my dissertation. Allison Schultz, Pascal Teitel, and Felix Ratcliffe, who helped me in Lassen. In Whitney, Teresa Feo was a big help doing surveys for me. Field assistants included Paul Newsom and Nadi Najjar, and Dan Wade. We got a squirrel. And then there's a whole lot of other people. Michelle and Carla have been huge helps throughout all the time. Michelle always answering all my crazy queries for data that I need now. Bill Monahan, who was a former student here and continues to be a collaborator of mine. The Beisinger Lab has helped a lot. Everyone who's ever worked on the GRP has helped me in some ways. And of course, MVZ and ASCOM have provided funding through some of the time. And thank you for listening.
We have some time for questions. Bill's got his hand up already.
It seems like one thing that's really missing are your analysis explicitly, although it's implicit, and that's the vegetation. And you're dealing with, you mentioned about primary productivity, but there's also structural aspects of the vegetation that can be important. So what are you going to do about the missing vegetation?
Well, the missing vegetation is something that's totally plagued us. We got our hopes up when we learned maybe 4 years ago about the VTM project and the Wieslander maps and the work that Jim Thorne's been doing at Davis and Maggie Kelly and Barbara Allen Diaz have been doing here. And we have, to a certain extent, and so the idea of that is there's historical Forest Service surveys that were done with detailed plot level data throughout the Sierras in the late 30s and early 40s. That to some extent has been gone out and resurveyed as well. The problem is the extent of that does not cover our survey areas. It covers Yosemite. It covers basically none of Lassen. There's this huge hole in the northeast quadrant of the state that has no vegetation data, which is very frustrating. And it covers about half of our southern Whitney transect. So I think in time there will be an opportunity to, at least on a fine scale in Yosemite, to maybe try and use some of the detailed vegetation data to see how it relates to the patterns of birds and mammals shifting that we found. But we just don't have the capability across the broad regional scale to look at that. I wish Grinnell had also taken, veg plots or something, but he didn't. We can't ask that much of him.
Yeah, go ahead, Dan.
So are there any reasons to think that changing fire regimes over the past 100 years might influence some of these patterns?
Certainly, if you look at historical, if you look at historical photos, and we've done a lot of photo retakes, you can see some evidence of possible evidence of shifting fire regimes or fire suppression, really, which is going to be changing the forest structure. It's hard to know how much. I mean, it's hard to know how much it plays. Yeah.
Questions, Jay, yeah.
So I'm wondering, I guess this is probably going to be a guess on your part, going on the vegetation data, but as a follow-up to Bill's question, do you sense that, or just what's your feeling, whether the shifts are a response that's sort of mediated by changes in vegetation, or do you think it's like actual physiological
I've almost gotten into fights over this. Because I actually believe that at least some species are going to have physiological limits that may directly, like limits in the breeding season, like with egg temperatures and frequency of severe cold snaps and things. Because we know from fine scale data that's that when you're looking at sort of early breeding season and things like that, the extent to which those things happen are changing how species elevational ranges, because species can change their elevational range relatively quickly. There's a lot of sort of facultative movements, particularly during the breeding season, at the start of the breeding season, trying to get highest as you can, because that's where the least predators are. At the same time as you can't basically have a nest failure if the entire clutch gets frosted. So I think there's probably some physiological limits. But it's going to be variable. I mean, certainly, probably not all the species we're looking at are going to be reaching physiological limits. But I think probably a certain portion of them are. I would love to test that, do fine-scale experiments. But yeah, it's a big unknown at this point. I mean, I'd say if it's not that, then, for instance, I think a lot of insectivores are probably going to be impacted by physiological limits on the invertebrates that they're eating, and that's actually going to probably be a bigger impact necessarily than changes in vegetation structure. That's my guess.
So maybe something I'm not fully understanding, but in the niche tracking, in the perfect equilibrium model, niche tracking means nothing changes in climate space, right? Because they move around to stay at under the same conditions. So I guess I'm a little puzzled what niche tracking means in relationship to. I didn't quite follow the method not related to on the ground.
For the colonization extinction part, right?
Maybe even before that, the vectors.
For the vectors, niche track. So you have. So it's whether or not basically this I need to put the vectors in. Whether or not this vector has the same sign as this vector. So in this case, the species, these arrows are both moving sort of negatively on the temperature axis and positively on the precipitation axis. So in a case where it would not be niche tracking, it would be moving that way.
What's the big cross? The gray.
The gray cross is the climatic niche for the species, and this is where we found the species historically in the Sierra Nevadas, and this is where we found the species in our contemporary surveys.
And the historical samples are put on the historical prism data, or onto.
Yep.
So I guess this is just a conceptual question. So perfect tracking, there should be no movement in climate space. I mean, if the species moves instantaneously to equilibrate with climate, and there's no mismatch of realized and geographic, you know what I mean? Then there should be no.
Yes. Well, it's different in some cases because you have to assume then that the climatic space that defines this range-wide niche is the exact same climate space available to the species in the Sierra Nevadas. And so pretty much these asterisks are always going to be off this to some extent because the species does not have the full niche available to it in climate space. And so what's happening over time is that different climate spaces are becoming available and different climate spaces are becoming less available. And so that's what's moving it in different relationships.
So for example, if you took the historical data and then said they didn't move and imposed it on the new climate space, you would get a vector.
You would, sometimes you would get a vector, yep.
What does that mean? That's not niche tracking, that's the failure to track.
Well, it means there's probably noise in there.
No, that's real. I mean, if the climate moves and the birds don't, you'll get a vector.
Yes.
And that's not niche tracking.
True, but that doesn't mean that it's moving towards it. You will net track it only if basically climate space is moving the Sierra Nevadas towards favorable climate. And towards favorable climate, towards where it is. A species doesn't necessarily have to, if you are in a good spot and you're being moved into a better spot, you don't have to move necessarily in order to track climate. the way we defined it, basically, we conflated this, what you might call active tracking and passive tracking.
Well, that definitely helps.
Okay,
But
yeah.
But I guess it still leaves me.
I would like to separate those two out, because you're right in that there's a difference between a species that has moved in order to basically actively track climate and a species that hasn't had to. We can't say much about the species that haven't had to.
But you should be able to unpack it pretty easily by taking the historical location data, put it on the modern climate. That's the passive part.
Yes.
And then and that's one vector.
Yes.
And the second vector is the new location data.
Yes.
And that hopefully would go from the blue to the red with a little point in between.
Yes.
And then it would be kind of self-think about what that would mean.
Yes.
Yeah.
Oh yeah, so it's just kind of closely related to that. I was wondering what time period was the Big Cross, where you took all of the occurrences over what time frame did that come from?
That's all pre-1940 in the historical data is pre-1940.
So I kind of expected when you first started showing this that you were going to use the difference between Grinnell, because that was roughly or closer to the same time period, I guess, as
yes.
The difference between Grinnell and the whole range conditions niche as sort of to calibrate any potential bias that. So at first I just assumed that the difference between the Grinnell survey and the range-wide just sort of represents bias sampling, the bias in sampling that, from, because Grinnell was only sampling certain areas, but that's not how you're using it. Anyway, I'm just wondering how you would be able to differentiate if the kind of whole range, yeah, I'm just struggling a little to tell what the difference between the Grinnell niche and the what are you calling it?
The range-wide climatic.
The range-wide niche, what that difference represents.
I mean, in some cases you could, like, it's a combination of, it probably, yeah, in some ways it is a sampling bias in the way that if you go back to here, this is what we were trying to measure. And this is sort of the center of this would be the gray cross in many cases. And it's the case in the Sierra Nevadas might be where we're sampling here. We can't, where the species is here and how it's moved is basically the best estimate we can get. Because we don't have any estimate necessarily of across the range where it is and how that's really, I mean, we have to define one as the climatic niche. And we can't, and basically this is the best we can do. And the best, so the best we can do then is look at where it is historically and how I don't know, that probably doesn't answer your question.
One more quick one, Michael, yeah.
I think we're talking about individual species differences, but have you considered using a phylogenetic framework to maybe tease apart maybe what's an individual species response versus the response to the genus or higher?
I haven't done any specifically like independent contrasts or any phylogenetic included mixed modeling. I've looked at it. If you look at it, there's not to the eye any really obvious differences, and certainly there's the conflation, the problem of the fact that you have a single species that is changing differently in your 3 different regions, and so what then do you put in as a single species response? So that makes it more difficult, and that's basically why we haven't done that.
It's about time. I can see next semester Morgan's going to be leading a seminar on niche tracking. Thanks a lot, Morgan.
Pose is the prefix pan, derived from that term pan monophylum. In addition, intermediate clades associated with the origins of various apomorphies are given names that describe the corresponding apomorphies. The main complication arises when the most widely used name also describes an apomorphy, and in those cases the name of the apomorphy clade is formed by combining the name of the crown with a standard prefix or suffix. The one that's been proposed is the prefix apo for obvious reasons. Here's an example using birds. The most widely used name aves or aves, is applied to the crown clade, that is, to the least inclusive crown clade containing all extant birds. The name of the corresponding total clade, which is formed by combining the name of the crown with the prefix pan, would be pan aves. It includes aves and all extinct species that share a more recent common ancestor with living birds than with any other living organisms. And of course, many of these species are also members of a less inclusive clade named Dinosauria. One of the apomorphies commonly considered important in the origin of birds is that of wings and flight. The clade associated with that apomorphy could be called aviali, meaning bird wing, or some other name that describes the apomorphy more specifically. Corresponding vernacular names could also be developed, and I proposed using the best known vernacular name, in this case birds, at least in English, modified with the appropriate adjective. So, for example, we would talk about crown birds for the members of the crown clade, pan birds for the members of the total clade. The intermediate apomorphy-based clades, for those clades we would use names such as feathered birds, bipedal birds, winged birds, short-tailed birds, toothless birds, etc. Another useful term is stem birds, which refers to the set of all organisms that are members of the total clade, but not of the crown. And of course, those are all distinct. In addition to increased nomenclatural precision, an integrated system of clade names has several advantages. One of them is cognitive efficiency. The system is efficient because knowing the name of a crown automatically means knowing the name of its corresponding total clade, and vice versa. Thus, for all crown clade total clade pairs, this situation effectively reduces the number of names that one needs to remember by half. A related benefit is that the integrated approach facilitates communication among biologists who study distantly related taxa. For example, currently, most botanists and entomologists probably have no idea what are the names of the total clades of even such familiar crowns as Mammalia and Crocodilia. In fact, I wouldn't be surprised if most vertebrate biologists don't know them. In contrast, under an integrated system, the names of these total clades would be immediately recognizable to anyone familiar with the names of the crowns. Yet another advantage of the integrated approach is that it results in a broader concept of origins, at least for distinctive clades of living organisms. Under traditional approaches, the best known names are commonly associated with particular apomorphies, which tends to focus considerations about the origin of the group on a relatively short segment of the stem lineage in which the particular apomorphy originated. In contrast, the integrated approach broadens the concept of origins to the entire stem lineage. Here's an example involving snakes. Under traditional views, as illustrated on the left, the origin of snakes is commonly thought of as the short segment of the stem lineage associated with body elongation and the reduction or loss of limb. However, snakes have many other distinctive characters relative to other extant organisms, some of which likely arose much earlier and others much later than body elongation and limb reduction. By deriving the name of the total clade from that of the crown, the integrated approach effectively treats the origin of snakes as the history of the entire stem lineage, as illustrated on the right. And this perspective has the added benefit of highlighting the wealth of transitional forms in groups with good fossil records, which seems particularly important in light of renewed attacks on the teaching of evolution. For example, in contrast with traditional views in which only those fossils close to the origin of wings and flight, fossils such as Archaeopteryx, are considered intermediate between reptiles in the paraphyletic sense and birds, the integrated approach highlights that there are literally hundreds of transitional forms. They include some extinct forms that are much more distant from modern birds than is Archaeopteryx, for example, sauropod dinosaurs, and others that are much closer to modern birds, such as the short-tailed but toothed Ichthyornis. And finally, another advantage of the integrated approach is that it would bring consistency to neontological and paleontological usage. The problem here is that although neontologists and paleontologists generally claim to use the same names for the same taxa, in practice neontologists tend to use the best known names for the crowns, ignoring less well-known names that have been proposed explicitly for those clades. For example, despite the explicit proposal of the name Lysamphibia for the amphibian crown clade, as illustrated here, neontologists almost always use the better-known name Amphibia when talking about extant species alone. Thus, we have names such as the Amphibian Ark, Global Amphibian Assessment, the Amphibian Conservation Summit, the Amphibian Specialist Group, Amphibiatry, Amphibiaweb, and countless books on the amphibians of geographic region X, all of which should really be using the name Lis Amphibia, since members of the STEM group aren't included in these works. However, if the best-known name Amphibia were to be assigned to the Crown, then all of those inconsistencies would vanish. So to wrap it up, an integrated approach to naming clades would have some important benefits. It would increase nomenclatural precision, and it would do so efficiently. It would broaden the concept of origins for distinctive clades of extant organisms, thus highlighting the wealth of evolutionary intermediates for many such clades. And it would unify neontological and paleontological usage. To achieve such a system, some concessions would have to be made concerning both the references of particular names and the names of particular clades. The result, however, would be a system that better meets the needs of biology as a whole. Thanks for listening.
So I know that some people are going to have to leave now who have classes, etc. So for those of you who'd like to go, you can file on out. And then I'm going to let Kevin handle his own questions since Greg's not here. But I want to mention also that for those of you who are interested, we're going to be going out to lunch afterwards, and anybody's invited to come along if they would like.
I had a question, which is, I mean, let's just say we all agree that clades are really important and that you should only have clades as taxa. I'm not sure that it follows that the name should actually be attached to clades. So, for example, when I explain to students the importance of clades, I say things like, look, fish aren't a clade, that's why Pisces is not a taxa, etc. Fish means exactly what you think it does, right? But if you attach names to clades, like, so for example, with reptiles, you might say something like, well, reptile means what you thought it did, it just turns out they're not a clade, they're not a taxa. Or you could say, which, if we had named reptiles 100 years ago or something, it might turn out that birds are reptiles, and that reptile still is a clade. But with some names that seems to me okay. Maybe I might accept that birds are reptiles, but for example, I definitely wouldn't think that we're fish, right? And so with your, like for example, your termite cockroach example, I think you're saying termites are cockroaches. So on the newer view, but another option it seems to me would be to say, actually, Blattodea, they are cockroaches. That means just what you thought. It's just they're not a clade, so they're not a taxa anymore. And
actually, while you're responding to his question, I was sitting here thinking, WWJD, what would Joe Belsestein do? So he would ask the following questions, and prefacing this by saying, all else being equal, I'm all in favor of things being named according to trees. But in his book, Inferring Phylogenies, he coined a new school, which is it just doesn't matter school. Right, and his argument was that practicing biologists who are doing this, using phylogenies, actually use the phylogenies correctly. The independent contrast is correct on the tree, or the family character is correct on the tree. So while you're addressing his question, you can deal with Joe's question as well.
Yeah, so in that case, I mean, I think, I don't disagree with Joe about that, but I think what he's overlooking is the fact that people do need to refer to things. And it's, instead of having to put up a tree every time and point to all the species in that clade, it's much more convenient to say mammalia or something. And so, I mean, I think he's sort of overlooking some things that maybe don't affect his world as much, but affect that of other practicing biologists more. Then on to the first question. I think there are several issues going on there. One of which is, first, there's a distinction between these formal taxon names and also just names that, there are lots of other names. Like there are all kinds of names that exist in every language that are not formal taxon names. And so this idea of attaching taxon names to clades is talking only about these formal taxon names. And it's following this tradition that goes back to Darwin of this idea that these taxon names should refer to strictly genealogical units or entities. So it's not to say that you can't use other terms, other names effectively, that don't refer to clades. And in fact, you even notice that when I was talking about the vernacular names that might be useful in referring to in the context of this integrated system, one of those vernacular names doesn't refer to a clade. When you talk about, for example, stem birds or stem mammals, that actually refers to a paraphyletic group. And people, I think, have taken that fact, people who've advocated this sort of everything is clades approach have taken it to the extreme that we shouldn't even use terms like that. But I think they're missing the point. I think the point is, no, maybe we shouldn't have a formal taxon name that refers to that group, but the notion of all those members of the total clade that are not members of the crown is still a useful term to have in certain types of discussions, and I think it facilitates. Well, if you think about it in the context of mammals, I think in the context of mammals, the alternative to that, to saying stem mammals or something, would be either to use a formal paraphyletic taxon. I don't know if there actually was one. There's some that refer to intermediate clades that are paraphyletic. Well, no, I think the name Synapsida originally referred to a paraphyletic taxon that didn't include mammals. It was the synapsid reptiles, for example. But if people were opposed to having a formal name like that, then their alternative is to come up with a very cumbersome name that probably nobody would recognize, like non-mammalian theropsids, something like that. And it's like, who knows what that is? Well, on the other hand, if you just say stem mammals, it's very obvious what that group collection refers to. Yeah, Chuck.
Yeah, actually, I think there's a little bit of a conflation here between words like reptiles and reptilian. And so reptilia is a formal name, but reptiles is not. And if you look at other kinds of common names like fish, fish is not a formal name, but neither is tortoise or turtle. And depending on where you are in the world, tortoise and turtle mean different things.